Good morning, everyone. My name is Lauren Carneal, and this summer I had the privilege of working under Dr. Greg Martin and Dr. Ning Zhang in the Martin Lab. I spent my 10 weeks looking deeper into the PTO and PRF disease-resistant signaling pathway in tomato plants. So first off, the bacterial pathogen that I focused on was Pseudomonas syringae PV tomato, or more commonly known as bacterial speck disease. This is a model system for understanding not only bacterial pathogenesis, but plant immunity as a whole. It is an economically important disease. As you can see in the pictures, there can be necrotic lesions on both the fruit as well as the leaf and the stems as well. This can pose a problem for tomato production as it negatively impacts the health of the plant, and it could decrease production and have fruits that are able to be sold. This is a disease that is spread through the stomata and open wounds of the plant, and it is just really something that decreases tomato production every year. So when a tomato plant is first infected, it goes through a two-layered immune response. The first layer of this response is pattern-triggered immunity, or PTI. And when PTI fails, ETI begins, or effector-triggered immunity. And that is what my project focused on. So I'm going to go a little deeper into the specifics of that now. To start off ETI, the pathogen comes to the cell and it secretes several effectors into the cell. I am focusing primarily on AVR-PTO and AVR-PTO-B. Once these effectors are in the cell, they get recognized. The receptor for this recognition is the resistance protein PTO and the kinase domain PRF. Once these two effectors are recognized, it triggers a kinase cascade, and this ultimately results in programmed cell death. Now, this is a very simplified version, and additionally, there was another lab at a separate institution led by Sophie and Kamoon, and they found these two proteins called helper NLRs. NLR is a nucleotide binding leucine-rich repeat, and NRC 2 and 3 are considered two of these helper NLRs. NRCs are simply NLRs required for cell death. It was unsure exactly how they fit in these pathways, if they are something that is more upstream during the activation process, or if they come in downstream during the signaling cascade. So that leads me to the objectives of my project. I first wanted to determine the importance of NRC2 and NRC3 in the ETI immune pathway, as well as investigate where in this pathway they exactly take place. To begin this, my mentor, Dr. Ning Zhang, developed a few CRISPR lines. Using a phylogenetic analysis, it was determined that NRCs came into 4 clades, and they were then separated to NRC 1, 2, 3, and 4. Using these clades, he created CRISPR-Cas9 mutants with a single knockout for the following. In addition, by crossing the single mutant for NRC2 and NRC3 and taking the F2 generation, the NRC2-3 double mutant was able to be created with both of these two NRCs knocked out. This leads us to the experimentation. First, I would like to talk a little bit about what disease assays are. This is a process used to completely inoculate the plant. They are placed upside down in an apparatus, exactly like this, and they are submerged in a bacterial solution. The air is then taken out of that chamber, and after a period of time when the hose is released, there is a dramatic change of pressure, which forces the bacterial suspension into the leaves of the plant, effectively inoculating the entire thing. This then was left in a growth chamber to grow for several days to monitor the health of the plant. 3 PST strains were used, DC3000, both with effectors AVR-PTO as well as AVR-PTO-B, and then DC3000 with only AVR-PTO-B and DC3000 with only the effector AVR-PTO. So right before I got here, Ning did an initial disease assay where she tested every CRISPR mutant she made. As you can see here in the wild type, also known as PTOR, this is the completely resistant line. It has a functioning PTO-PRF gene and can have no susceptibility to PST. This contrasts the negative control, or PRF3, which has a deletion in the PRF gene, making it completely susceptible to this pathogen. Now, as you can see, right next to PRF3 is the NRC23 double mutant, which also showed a complete and total susceptibility. This is really interesting. As I mentioned the Commune lab earlier, they did a similar experiment, but with Enbenthiana, and they saw similar results, but they did a transient expression where now this is a complete knockdown of the genes responsible. And then after that, we noticed the single mutants all seem to have overall resistance. So this started to beg the question if they act with AVR-PTO and AVR-PTO differently, now that we know NRC2 and 3 are both very important in this process. So this is where my research started this summer. I conducted a disease assay using DC3000 with AVR-PTOB without AVR-PTO. And I kind of got pretty similar results to the initial study. As you can see, NRC23 double mutant did have complete susceptibility. And overall, the single mutant NRC2 and NRC3 seem to be overall resistant. However, if you look a little more closely, the NRC3 knockout plant seems to have an increased susceptibility to this disease as compared to the NRC2. This indicates that possibly NRC3 acts more strongly in this resistance pathway, and while they are both required and may be redundant, they are partially redundant with different interactions and different strengths. This is also mirrored in a bacterial population load as shown over here. As you can see with the yellow bar, that is the double mutant, and it matches the bar to the right of that as the susceptible PRF3. And then the gray bar is much taller than the orange bar, showing that NRC3 did also have a quantifiable difference in the bacterial population load, making it more susceptible to this pathogen. This was repeated in DC3000 with AVR-PTO, and once again, similar results were found, showing that NRC2 and 3 are required as recognition for both of these effectors. Once again, you can see an increased susceptibility in the NRC plant, which is once again reflected in the bacterial population load. So now that we've figured out that these are definitely important in ETI, we wanted to look at where exactly in the pathway these two proteins come into play. And this was done by completing some cell death assays. Cell death, as I mentioned earlier, is one of the end results of ETI. This is simply a physical manifestation of the immune response of the plant that we can see the death. To do a cell death assay, the leaf was first pricked to create a wound, and a syringe containing an agrobacterial solution was pressed into the leaf, creating the solution spreading and therefore inoculating as seen in this picture below. And it spreads out into a more controlled area to handle the infection more than just the whole plant as seen in the disease assays. So to do the cell death assays, I use several programmed cell death inducers. I use the autoactive proteins of AVR-PTOB, PTO, PRF, MAP3K, and MAP2K. These are reflective of the simplified pathway over here and shows the order of the cascade. These proteins are constitutively active, which means that they do not require the pathogen to activate, and we will still see a response without an infection of Pseudomonas syringae. So, as I mentioned earlier, PTO is our resistant cultivar, and it is hypothesized that it will have positive cell death at all stages of the ETI response. PRF3, or our negative susceptible line, should only have cell death at the end of the cascade for PRF, MAP3K, and MAP2K. And we are trying to figure out where exactly NRC2 and 3 come in. So we weren't really sure what to expect. And this just kind of emphasizes, once again, we don't know where exactly they are in the pathway, if they're above the cascade or if they come in a later downstream. So this brought us to the results of the cell death assay. First, we looked at PTOR. The pictures indicated by the red box are positive for cell death. This was what we predicted. As you can see at the end, this is YFP. This is simply our negative control. It is an empty vector. It should not elicit cell death. The wounds that you do see on there are caused just from the syringe. It is just a process that wounds the plant, as well as the bacterial strain may cause auto lesions. But this cell death is nothing compared to the other samples. So then we looked at PRF3. And once again, we got the expected results with only cell death in PRF3, MAP3K, and MAP2K. So now that we know that our assay is properly working, we're going to take a look at what happens to NRC2 and 3. NRC2 and 3 had only obvious and significant cell death in MAP3K and MAP2K, and the other samples had little to no death. This was really interesting and exciting, as now we're kind of wondering, does it really just come up here above the MAP kinase cascade? It could be involved in a complex with PTO or PRF, or in some other type of signaling. Further experimentation will have to be required, but now we know it is most likely above this cascade in the process of ETI. So this leads me to an overall summary of my project this summer. For determining how NRC2 and 3 act in an ETI response, I conclude that they act partially redundant, with NRC3 being the stronger of these two. And as for where they are in the cascade, it is hypothesized that they are upstream of MAP3K and MAP2K in the entire MAP kinase cascade, and they may possibly be in a complex with PTO and PRF. This leads us to what's next for the Martin Lab in continuing this project. There should be an additional replication of the experiments performed this summer just for an increased confidence of the results. An examination of the PTI response. This, as I mentioned earlier, is the first fold pattern triggered response. And while NRC2 and 3 are involved in ETI, it should be interesting to see if they are involved in any sort of PTI response as well. A phenotypic and growth development, taking a look at that could also be interesting. I mainly worked with 4-week-old seedlings when inoculated, so it should be interesting to see how these lines develop into fruit and flowers as they grow and get older. And then finally, an investigation of how NRC2 and NRC3 overall act in this pathway on the more molecular level, if they do in fact act in a complex with PTO and PRF, or if they are involved in some other way. I would like to thank the entire Martin Lab, especially my mentor, Dr. Ning Zhang, as well as my PI, Dr. Greg Martin, as well as everyone who made this RAU and the funding possible. If you have any questions, feel free to reach out to me. Thank you.